So in the interest of time, we're going to try to get started here in just a minute. So I'm Zachary Hunter, and along with a good friend of mine, Dr. Varretoni.
Good afternoon. My name is Marzia Varretoni. I work at the Policlinico San Matteo in Pavia, in Italy. I'm very happy to be here with you. It is the first time for me at the Patient and Physician Summit, and I'm really, really happy for this. I let Zachary start with the first part of his presentation. I will follow him with a clinical point of view, as I am mainly a clinician. So, Zachary, you can start.
So I'm going to mostly be focusing on stuff that came up and try to cover everything that came out of the session that just happened, actually over the last couple of days here in New York. So it's a lot of stuff. It's a little fast. I apologize in advance. Really ask questions. Feel free to ask questions, and yeah, hopefully this will help. So before we get started, we keep talking about these mutations, but I wish we had more time to go into some of these details, since I know when I first started researching, I hadn't taken biology since high school, and I was a little confused at the time. And I was like, wait, I remember DNA, something. But basically, what everyone should know is that DNA is not the only thing that we inherit from our parents, but it is the major thing that we inherit. And it's big. We have about 3 billion base pairs that we get, one copy from each parent. That's a lot of information, right? And it's not, we used to, if you go back to the 80s and you look up things, there's all these twin studies about separated at birth, and they like the same toothpaste. It turns out if you take 2 random people on the street and you just ask them enough questions, you're going to get stuff like this. And it's not to say that identical twins are not incredibly similar and that DNA is not important. DNA is incredibly important. But it is really more of a blueprint. It is not the determination of your life. It is not who you are. And as a matter of fact, follow-up twin studies when they're much, much older, they're quite different from each other. They like different things. They have different experiences, because in reality, who we are is shaped by our decisions, our interactions with our environment, and so it's bigger than all that. Now, that being said, DNA is also really important to cancer, and so what happens is that as we age, mistakes happen, changes happen in the DNA, and often it's fine. But when enough of them accumulate, you get this problem where the code in the DNA that's telling your cell how to do different things gets damaged enough that it starts to lose some function. Or sometimes it gains the function that it didn't used to have. And when enough of these accumulate, right, you can get out of control growth. Like a cell that used to know, hey, I'm supposed to stop growing now, doesn't know that anymore and keeps on going. And that's really what we start calling cancer. And then so the big issue now for all of us is if cancers are arising from our own cells, how do we figure out, how do we target these cancer cells without targeting ourselves? And that's oncology. That is what we're all up to. And my job as a researcher is to try to understand the biology so we can define new targets to be able to do just that. So that's what we're going to cover a little bit in this session. So back in 2013 and 2012, Steve and I published a series of whole genome sequencing where we went and we looked at all 3 billion base pairs and found actually there were a number of genes that had been recurrently mutated across WM patients. And the most famous one is MYD88, but also CXCR4, CD79B, and TP53. So these are genes that we're going to talk about in this session a little bit. The most common ones are MYD88 and CXCR4, which I think we've been hearing about over the last few sessions. But these other genes also play an important role. Now, this initial screen was not that sensitive, so we've now gone back with better techniques, and we can say that actually the MYD88 mutation rate is actually well north of 90%. CXCR4 rates are now probably about 40%. using more sensitive technology. So what is MYD88? MYD88 is actually part of your normal immune system. So it's, your immune system, you could say, is divided up into two parts, the smart part and the not so smart part. This is part of the not so smart part. There's a reason why you don't get the same disease twice, right? Your body learns how to fight something. That's the smart part. But the problem is it takes a little while to kick in, right? So while you're waiting for it to kick in, you want something really fast, right? Hey, you have an infection, let's get on it. And that's where these toll-like receptors come in. They don't know that much, but they're really good at picking up things that are super common on bacteria and viruses, right? So there's certain proteins and signals that are on bacteria and viruses that these are really good at picking up. So it doesn't know what's there, but it knows something that's not supposed to be there is there, and tells the immune system to freak out. And so MYD88 is actually the control for this. So when these receptors go and they pick up one of these things, it causes MYD88 to come together, two different molecules of it, and propagate this downstream signaling. And what happens in Waldenstrom's is there's a mutation that causes that signaling to be on all the time. So it's always seeing bacteria, it's always seeing viruses, it's always sending out this inflammatory signal. Hey, we're seeing something, we're seeing something. We need to go fight this. And hey, maybe help me out too, as long as we're at it. And so this activates is a very important molecule called NF-kappa B. And you don't really need to know too much about that, other than the fact it binds to the DNA and sets forward a transcription factor or a transcriptional program, a signaling program within the cells. That's just really, really important for their survival. Now, Guangyang and our group made an additional really important discovery, and that is that MYD88 doesn't just signal down through this canonical pathway, it also signals through this molecule called BTK. Now, BTK, as I think many of you know, is the target of Ibrutinib. So the vast majority of the signals from MYD88 go through BTK, and when we had this idea of treating people with Ibrutinib, that's what we're trying to target. We haven't figured out how to target MYD88 directly, but we can go after BTK and therefore shut this pathway down. Now, with CXCR4, it's a little bit more complicated. We see a whole series of mutations. So in MYD88, you're going to hear a little bit more from Marzia. It's really more or less one mutation with very rarely a couple other ones getting involved. In CXCR4, it's just all over the place. But if you look at those red boxes, that's sort of it looks like gobbledygook, but believe me, it's actually a protein code. It's all towards the end. All those mutations are right there at the end. And the reason is, and you can look here, if you, you see the picture with the big arrows on either side, there's something that looks like handlebars almost. So the main part of the protein sticks through the cell surface and acts like a receptor. Those handlebars are there actually to help turn it off. So when the receptor is activated, on some sense, the cell grabs hold of those handlebars and pulls it back inside, and that turns it off. So what happens here is we get mutations that either cut it off entirely or scramble it, so the cell doesn't know how to grab on anymore, and then the cell can't pull them back in. So they just sit there on the surface, and they signal, and they signal, and they signal. And so what does this do? This is actually something called a chemokine receptor, and it's the GPS sort of for a cell. It helps cells know where to go. And in this case, it's helping it go to the bone marrow. And you'll hear these occasionally referred to as WM-like mutations, and that comes from a germline, it's an inherited disease that is unrelated to Waldenstrom's, but has the exact same types of mutations. And what happens is that all of their immune cells home to the bone marrow and refuse to leave. And so for this reason, they have hypogammaglobulinemia, which probably sounds familiar, infection, warts, which luckily, hopefully doesn't sound that familiar, and also something called myelokathexis, which is a fancy term you don't need to feel bad about not knowing because they actually invented it for the disease. So there are some clinical things that go along with this, and again, Marzia is going to talk a little bit about it, but with the idea of sort of form following function, not surprisingly, people who have these mutations don't typically have lymph node involvement. They don't typically have big spleens because these mutations are telling them to go to the bone marrow. So we really see that more in this sort of CXCR4 unmutated population. However, with people who have those sort of truncating mutations in CXCR4, we do see the highest levels of IgM and bone marrow. And also across all of these CXCR4 mutations, we also see this, this sort of therapeutic resistance to certain therapies. And it's just something you should know. On the other scale, we have people who don't have the MYD88 mutation. This is a pretty small group. But they do exist, and they are really important. And they typically actually have low bone marrow involvement, low IgM. And then clinically, and Marcia's going to go over this more, just they also typically, like when you look at overall survival, and I don't want to focus on this too much, you'd think the CXCR4 people would be really scary with the high bone marrow and high IgM, but they're not. Actually, we don't really see much of a difference there. It's the people who are wild type or normal for MYD88 that we really see having a harder course of the disease. And in part of that, that's because they transform into a more aggressive type of cancer at a much higher rate than the rest of WM. And we'll talk about more of that in a minute. And then finally, we recently started to look into this, what it is that makes these MYD88 wild types different. What are the mutations that are driving them? So if MYD88 is driving you sort of more typical WM, what about these other people? Well, we've looked into it, and there's no need to write down all the different gene names or anything. The only thing I really want people to understand is that everything sort of there on one side of the graph is starting from TBL XR1 all the way over to the UDF1L. Those are all genes associated with NF-kappa B. Now, if you remember, NF-kappa B was the endpoint of the signaling for MYD88. Now, this is doubly important because we had recently described that people who didn't have the MYD88 mutation didn't really respond to Ibrutinib that well. Right? Now, if you remember, Ibrutinib's targeting BTK. BTK is right downstream of MYD88. And when you go and you look here, you see all these little triangles. Those triangles are where the mutations, activating mutations are in these MYD88 wild type people, and you'll see that they're all beneath BTK. So inhibiting BTK doesn't stop these mutations at all. So we're still working to understand the best way of doing this, but we probably are going to need a different therapeutic strategy for these individuals. And so this is something that we're actively working on. And the last thing I want to just talk about very briefly, and again, don't worry about gene names, we see dysregulation of genes related to cell survival. So cells very carefully manage whether or not to live or die. And that might sound like a strange thing, but cells get damaged. And sometimes they get damaged to the point that it just doesn't make sense. It's better to have the cell die and be replaced by another cell than to try to make it work. And so this is carefully managed. And one of the things cancers do is they abuse the system and they crank up the pro-survival genes. And indeed, that is what we see, at least in MYD88 mutant WM. And I bring this up really just to give you a big picture about why we do this, right? Because when we understand what's going on, we can try to devise targets. We can then try to devise therapies for this, right? You know, we have Ibrutinib for BTK. You're going to be hearing about new next-generation BTK targeting compounds, Zanubrutinib, Acalabrutinib. We have venetoclax, which is a BCL2 inhibitor. This is targeting this pathway that's controlling cell survival. So we know that this is a problem. We can now design drugs that go and target that pathway and can overcome that problem. And finally, we can target CXCR4 itself. So there's a clinical trial that's now underway at the Dana-Farber using a drug that's impossible to pronounce. It's called ulocupilumab. Who paid for that name? But it blocks CXCR4 activation, right? And so we're now using it in combination with Ibrutinib to see if we can help improve the responses to Ibrutinib for the CXCR4 mutant people. You'll be hearing again more about that CXCR4, the role of CXCR4 in Ibrutinib therapy, I think, from Marzia a little bit later. So I wasn't the only person who spoke. I want to cover some of these other presentations very briefly. One of the questions was, what about 6q losses? What is 6q is also a completely valid question. So your DNA is broken out. It's not one big strand of 3 billion things. Luckily, it's broken up into pieces. There's 23 pieces, 22 that are everybody has, and then there's a sex chromosome that's a little bit different between men and women. Women have 2 X's, men have an X and a Y. But if you're familiar with 23andMe, that's what they're referring to. They're referring to the 23 chromosomes found in humans. So 6 is the 6th largest chromosome. That's all it means. And they kind of look like airplane propellers, since we talk about the short arm and the long arm between the thing. Q is always a long arm. Doesn't matter. The main thing is that we see it deleted a lot in Waldenstrom's. And actually, it's not even unique to Waldenstrom's. We see it in a lot of lymphomas. So what's going on? And so this is data from our whole genome sequencing study that I mentioned earlier. And you can see that some of these deletions are really, really strong, really clonal, go throughout all the cells. Some of them are barely detectable, only in a few cells. But there's really a lot there. And so I worked with, actually, one of Marzia's trainees, who is wonderful, and she sent over to us. And we all worked together to study this more formally. So Maria Luisa Guerrera went and looked at 5 genes in 6q and tried to really say, okay, how frequently is this gene deleted? What percentage of cells is it deleted in? And can I learn anything about this? And so what she discovered, and you can kind of just almost see it clearly here, which is that there's a group of people for whom all the genes are deleted, and it's just deleted in all the cells. At least one copy is. And then there's another group of people where you get a deletion here, a deletion there. They're typically only in a few cells. It's not quite as dark blue. And the interesting thing about this is that we only see these like big deletions in people who don't have CXCR4 mutations. Interesting. People who do have CXCR4 mutations, we don't really see this. And then equally interesting, when we look at people who have asymptomatic versus symptomatic Waldenstrom's, we see 0 differences. So we see this sort of mutual exclusivity between 6q deletions and CXCR4. And so whenever you see something like that, it should make you just pause for a moment and be like, okay, that's got to mean something, right? So we went back and we looked at our transcriptional data, this is how the genes are being used, and looked for genes that were really strongly associated with deletions in 6q, right? And normally when you do this, you can figure out ways of visualizing how the cells relate to each other. And you expect all the samples deleted for 6q to be on one side and all the ones that aren't on the other side, right? I mean, we picked out the genes such that this would happen. But once in a great while, you wind up with a third group. And this is what happened. We got the first 2, and then we got the people with CXCR4, who are there in green below, as this sort of intermediate signaling type, which was really weird because we didn't choose anything to select these people out. And what we discovered was, in fact, there is a subset of genes that are modified in the exact same way by both events. If you have a CXCR4 mutation, these genes are modified. And if you have a 6q deletion, these same genes are modified in the same way. So this is important because when we've been trying to piece together how do you go from MGUS to asymptomatic WM to full-blown WM, we see MYD88 mutations, we see CXCR4 mutations as early as the MGUS clone, right? And if they're doing that, and we already showed that asymptomatic versus symptomatic WM, we don't really see any differences in 6q. So this is really helping us place these events sort of in this MGUS to smoldering transition, and also these genes that we were able to identify, therefore, as being really important in that transition. So that's a really exciting time. People are doing a lot of research to try to figure out what that is and see if we can target it and see if we can prevent progression, for instance. Or if there's still dependencies, these cells still depend on some of the pathways that allowed this to progress forward. So one more study I want to talk about just briefly is one by Ruben Carrasco, who talked about mouse models introducing human mutant MYD88 to mice to see if we could create a model of Waldenstrom's in a mouse. And with the sub-question being, is it enough? if we give these mice mutant MYD88, will that give them Waldenstrom's? So it's tricky how you do this. You first have to introduce it to a mouse, and that's its own process. We don't have time to go into it. But then, you don't want to just give MYD88 everywhere. We don't have mutant MYD88 everywhere. We have it in B cells, right? So how do you do that? So what you do is you insert a little special piece of DNA right in front of the gene. So right in front of your MYD88 gene, you put a little stopper in that prevents it from being used. It's totally useless. Doesn't do anything. But it's a special kind of stopper that if you expose it to a protein, it'll delete itself. And then what we do is we have another mouse that will produce that special protein, but it will only produce it in B cells. And then we cross these two mice together, and then just like that, you have a mouse that will make mutant MYD88 just in their B cells. So that's what he did. And the first thing we noticed, interestingly, is that they got these neck rashes. And at first, we were like, we have no idea what to do about this. This is really strange. But then what's interesting is just this year, there was a case where a person actually had juvenile rheumatoid arthritis, like really young, really bad, and then also developed this very severe neck rash. And they went and they sequenced it, and it turned out that she was born with mutant MYD88. So it turns out when you copy 3 billion base pairs, actually remember you're doing it twice because you have one copy from each parent and only one of them has to go wrong. You're actually doing it 6 billion for each cell. So let's say, let's start with an egg and going out to like 100 million cells. That's not a lot of cells, right? So that's 100 million times 3 billion base pairs of DNA, and you're not going to make one mistake? No, mistakes happen. Mistakes happen. They happen a lot. Most of them don't matter. But every once in a while, you'll get, it's called a de novo mutation. It only exists in the person, but it's still throughout their whole body because it's a very early event. And this is what happened here, and she got this very, very similar neck rash. So we got very interested, and we started characterizing these more, and Dr. Krasko was able to show that they developed what we call a polyclonal lymphoproliferative disorder, which means that their immune system was growing a little bit too much, their B cells in particular, and they were making a lot, as it turns out, of IgM. With additional follow-up, 4 of those mice went on to actually develop full-blown lymphoma. And what was interesting about this in particular is that those cases which had actual lymphomas develop also had high levels of a cytokine called CXCR4. And this is a cytokine that we had previously associated with Waldenstrom's. It's very, very high in Waldenstrom's patients and correlates very strongly with bone marrow involvement. So they're now planning to actually update this. They're trying to develop a 6q deleted mouse and to cross in with this MYD88 mouse to see if we can get these to develop more like WM, maybe also develop a little bit faster. Which also, if I can tell you, if you're ever like minding your own business, sitting at your computer, and Ruben Carrasco comes by and asks if you're doing anything, the answer is you're very busy, or otherwise you're going to spend the rest of the day trying to figure out what's the equivalent of 6q on a mouse. It turns out it's on chromosome 10. So I'm not going to talk about TP53 mutations, but there's a wonderful presentation by Stéphanie Poulain from France on this topic that Marzia is going to talk about a little bit later. And, but I am going to talk just very briefly again about transformation. So this is another presentation from Cristina Jimenez and Ramon Garcia Sanz from Salamanca in Spain. And they're asking the question, and I think you've probably heard about this, is in a it's not a super common thing, but Waldenstrom's can gain additional mutations that will cause it to go to another more aggressive disease. Now this transformation is typically really bad. It's really aggressive form of a disease called diffuse large B-cell lymphoma. So how does that happen? And can we predict in advance who it's going to happen to? And if we can understand it, again, with the idea, can we prevent this? So yeah, it's poor prognosis. It's it's a rare cumulative incidence at 5 years, 1%. So the goal here is just to define biological risk and see if we can, intercept it before we actually have the full-blown problem of DLBCL. Because once we're there, then it's much more complicated. And so Christina is trying to do something that's very complicated, which is that, we talk about these mutations, but actually as tumors grow, they keep developing new mutations. They don't stop changing. They keep evolving. And some of these mutations never grow out to do anything. They're there, doesn't mean much. Other ones will found whole new clones, and whole new versions of WM will be based on that. So a way to think about that is you might start off with a MYD88 mutation. And you might go wrong for a little while, and then CXCR4 mutation, right? That now allows the cells to survive a little bit better, resist certain kinds of therapies, go along, get a different kind of mutation. And so each of these are what we call clonal events that will cause sort of a new branch in this evolutionary tree of the tumor. And so what you want to do is you want to be able to select out those which aren't going to go anywhere to those with, like this one here circled, versus this one which is going to go on and actually found, for instance, in this case, a DLBCL clone. And so after going into this and looking into it very much in depth, she was able to show actually something similar to what we had seen, which is that people who are MYD88 wild type, and they have no MYD88 mutations, have a higher rate of transformation. We don't really know why. But for people who have the MYD88 mutation, there's this other gene called CD79B, and those people who had the mutation had a much higher rate of transformation to DLBCL. Now, to be clear, not everyone who has the mutation goes on to get this, but there's a clear association between the two, and this is interesting because CD79B is very common in diffuse large B-cell lymphoma, as is in the same subtype where you see CD79B, you also see mutant MYD88. So at this point, I'd like to turn over the platform to my good friend and colleague.
As I said before, I am very happy to be here and to co-chair this session with Zachary Hunter, because as you have seen, Zachary Hunter, together with Stephen Treon and the other members of this group, have provided most of the knowledge that we have so far on the biology of this rare disease. As I said before, I am mainly a clinician, closer to the bedside than to the bench. And so my task will be to show you how these recent discoveries made in the laboratory are changing and have already changed the practice of clinicians and the cure of patients. The starting point is, of course, the discovery of the MYD88 mutation made by Stephen Treon in 2012, when he found that more than 90% of patients with Waldenström macroglobulinemia harbor this mutation, which has become an important tool for us in the diagnosis of Waldenström macroglobulinemia, but has also an important role as a prognostic marker, either in patients with IgM-MGUS or patients with Waldenström. Will probably become important also in the molecular assessment of response after treatment, especially in the era of new drugs. And finally, and above all, the most important things for you, the discovery of this mutation has paved the way to the development of new drugs targeting this pathway, the MYD88 pathway. Let's start from the diagnosis. After the pivotal study published by Treon in 2012, other studies in different parts of the world have confirmed that the MYD88 mutation is highly recurrent in patients with Waldenström macroglobulinemia. But these studies have also demonstrated that this mutation is detectable at least in a half of patients with IgM monoclonal gammopathy of undetermined significance, the benign condition that may precede the onset of Waldenström macroglobulinemia. Unfortunately, the mutation is not completely specific for Waldenstrom macroglobulinemia. Different studies have shown that this mutation may be found in a minority of patients with other lymphoproliferative disorders, such as splenic marginal zone lymphoma, B chronic lymphoproliferative disorders not otherwise specified, or chronic lymphocytic leukemia. That's why, unfortunately, to date, the diagnosis of Waldenstrom macroglobulinemia and its differentiation from other lymphoproliferative disorders still relies on bone marrow findings. But we hope that in the future something may change, and and the bone marrow assessment, which is not fair and which is not simple to do for you, will be not necessary at all. What about the prognostic role of the MYD88 mutation? In this workshop, I showed some data from our group in Pavia, because I was asked to answer the question whether the MYD88 mutation is associated with the risk of progression from the monoclonal gammopathy of undetermined significance to Waldenstrom macroglobulinemia. And we found that using allele-specific PCR, the mutation is detectable in half the patients with IgM-MGUS. Probably using more sensitive methods, we will find that a higher percentage of IgM-MGUS patients harbor the mutation. But with the method we used, half the patients had the mutation. The risk of progression for the whole cohort of patients we studied, that were 193 patients, was 1.2% per year. And the most important thing, the most important finding is that the presence of the MYD88 mutation is a predictor of progression to Waldenstrom macroglobulinemia or to another lymphoma, irrespective of the size of the monoclonal protein. By combining the presence of the MYD88 mutation and the levels of the monoclonal protein with a cutoff of 1 gram per deciliter, we have demonstrated that patients who have the MYD88 mutation and have a monoclonal protein of at least 1 gram per deciliter have a higher risk of progression as compared with patients with none or one of these two risk factors. And this allows us to identify a subset of patients corresponding to a quarter of our cases, which deserve a more accurate, more careful follow-up. What about the prognostic role of this mutation in Waldenstrom? Here, the scenario is different because the majority of patients, who are those who harbor the mutation, have a better outcome as compared with the minority of patients who do not have the mutation. And as brilliantly shown in this work by Garcia Sanz, probably this reflects a different biology of the disease. And this group of this subset of patients who are MYD88 wild type is probably deserves further studies in order to understand their biology and to give also to these patients more effective treatment. What about MYD88 and histologic transformation? The histologic transformation, which means that Waldenstrom macroglobulinemia, which is an indolent lymphoma, became an aggressive lymphoma, is fortunately a rare phenomenon in Waldenstrom macroglobulinemia. We know that this aggressive transformation may occur in other lymphomas, especially in follicular lymphoma. In Waldenstrom, this rarely happens, and the estimation is that it happens in 2% of patients after 10 years from diagnosis. Here again, MYD88 assessment is important because TREON has demonstrated that those patients who are MYD88 wild type have a higher risk of transformation as compared with patients who harbor the mutation. What about response assessment? In this meeting, a young colleague of mine has shown our data on 33 patients harboring the MYD88 mutation who were treated with immunochemotherapy in our institution. And for these patients, we had bone marrow samples before and after therapy, and we assessed the MYD88 mutation status before and after treatment. What we found is that approximately 60% of patients became, in 60% of patients, MYD88 was no longer detectable after treatment. And most importantly, these patients seem to have a better outcome, a longer progression-free survival as compared with patients who do not become negative. This confirms a concept that is known in lymphoma, that the more the response is deep, the more it is predicted to last over time. Finally, the most important thing for you, the MYD88 has become a therapeutic target. The study that led to the approval by the FDA and subsequently by EMA of Ibrutinib was published by Stephen Treon in 2015. And this study was conducted on 63 patients with relapsed refractory Waldenstrom macroglobulinemia, showing that it is possible to target the MYD88 pathway using this oral drug, which binds to the Bruton kinases. This drug had already been approved in mantle cell lymphoma and in chronic lymphocytic leukemia. And to note, it is the first drug that has been approved for Waldenstrom macroglobulinemia more than 70 years after the discovery of the disease. What about the remaining genomic landscape? Zachary has already shown you the other mutation that he has found in a study of whole genome sequencing. I will focus only on two mutations which have important clinical implications. The first one are the CXCR4 mutations that, as you can see, may be found approximately in 25 to 40% of patients, according to the assay used for the assessment of the mutation. And these mutations may be present also in a minority of patients with IgM-MGUS. As Zachary has said you before, the presence of CXCR4 mutation is associated with a peculiar clinical presentation characterized by higher IgM levels that led to a higher incidence of hyperviscosity. There is also a higher, more extensive bone marrow infiltration, and that's why patients are more frequently cytopenic. On the other hand, the CXCR4 mutation are associated with a lower frequency of adenopathies. In a study published last year on asymptomatic Waldenstrom patients, we found that the presence of the CXCR4 mutation was associated with a shorter time to treatment, a nearly need to treatment, because patients tend to become symptomatic earlier. As Zachary has said, the presence of CXCR4 mutation is also an important predictive factor for response to Ibrutinib, because patients who are MYD88 mutated and do not have CXCR4 mutations are those who respond in the best way to this drug. Patients who are MYD88 mutated but also have CXCR4 mutations tend to have lower response rates and a shorter, delayed response and shorter progression-free survival with this treatment. But Zachary has already told you that fortunately there is an ongoing trial combining Ibrutinib with ulocuplumab that has the objective to increase the response rates also of patients with MYD88 and CXCR4 mutations. The subset, the small fraction of patients who are MYD88 and CXCR4 wild type is the subset that has the worst response to this drug, because these patients achieve only superficial and short living responses. But this confirms again that these patients deserve appropriate and dedicated clinical trials for achieving better results. Finally, a few words on the TP53 mutations. These mutations in other lymphoproliferative disorders have a well-known unfavorable impact. And Stephanie Poulain has published last year a paper where she demonstrated that these mutations occur in less than 10% of patients with Waldenstrom macroglobulinemia. She found that the outcome of patients was worse when the TP53 mutations are present, and these patients usually have a shorter time to treatment and a shorter time to progression after treatment. However, as Joshua Gustin has demonstrated in this paper published this year, the TP53 mutations are rare and are responsive to treatment with Ibrutinib. To summarize, significant advances have been made in the last few years on the biology of Waldenstrom macroglobulinemia. Some of the genomic alterations that have been found in Waldenstrom, such as MYD88, CXCR4, and TP53, has important implications for diagnosis, prognostication, and treatment decisions. These developments are paving the way to even more personalized treatments, which may into account patients and disease characteristics and also genomic profile. As you can see, in the past, we thought that one disease had the same treatment for all patients. Now we know that every patient is different from another, and when we choose the treatment for that patient, we have to take into account many variables that include the age of patients, his performance status, how the disease presents, and how, for example, how the patients need a rapid control of disease. The genomic profile, as we can see, as we have shown, is an important determinant for treatment decisions, but there are other variables such as comorbidities, concomitant medications, and also patient's preference. So the decision is not easy because we have to take into account the whole picture. Finally, I would like to thank all my colleagues, not only in Pavia, but here at the Dana-Farber Cancer Institute, and also other colleagues in Italy and in Salamanca for ongoing projects, ongoing collaborations. And above all, I would like to take the chance to express my gratitude to the International Waldenstrom Macroglobulinemia Foundation. I have the privilege to meet here the president, and I am really grateful to this association, because the International Waldenstrom Macroglobulinemia Foundation is funding an important project that has just started in Italy and in Spain, which is very complex. I don't want to go into details, but it has an important objective in terms of diagnostic. We want to see if it is possible to make the diagnosis and to follow the patients without the need of invasive procedures. We want to see whether it is possible using very sensitive methods to evaluate the minimal residual disease after treatment and to understand how the minimal residual disease negativity impacts the progression-free survival. And we want also to understand how the cancer changes over time during the natural history of the disease, often after as an effect of treatments. So it is a very important project, and I'm really grateful for the support. Thank you very much.
So I think we have time for a few questions.
Hi. I was wondering about the specificity of B cells. So do you see MYD88 mutations in germline that would cause also T cell mutations? And if so, what happens? Or is there a parallel universe of T cells with MYD88 mutations with their?
I've never heard of that happening in T cells. And the germline case that I mentioned was it's not inherited. It's something that happens specific to that person during their development.
So then why wouldn't it, I mean, MYD88 is in B cells and T cells. Why would it only be in, how do you explain the specificity?
I would say probably the biology. I mean, obviously we don't know, right? There's a lot we don't know. But my guess is there's something specific about B cell biology that makes this an attractive target for the development of cancer in B cells as opposed to T cells. But do you have any?
No, I fully agree with you. We don't know a lot of things, but we don't have experience that the mediated mutation is also in other cells.
Right.
I mean, I would predict that a MYD88 mutation in a T cell would cause autoimmunity. And maybe that's part of the disease, I don't know, in some patients, like the MAG.
Again, it's never been observed. I mean, but let me reframe the question in a completely different way. So, you know, we have been, and not just in Waldenstrom's, in a lot of indolent B-cell lymphomas, we have been unable to eradicate the disease, right? Pretty much full stop. There's a couple of people maybe in the history that we think we might say it's never coming back, but we don't really know. That being said, as hard as we work, we can't keep the cells alive outside of a person. So if we can't kill them in a person, we can't keep them alive outside of a person, we clearly have a lot we don't understand still about the biology, and I think this sort of falls into that category. My guess is when we more fully understand the role, it'll become clear why we don't see it in T cells.
Zach, I think it was in Phoenix that there was discussion about detecting MYD88 in peripheral blood, and if I remember correctly, you had some success, but there were false negatives, I think was the situation. Has there been any development since? Is there any chance of that being used clinically, or is that really the purpose of your research?
Yeah.
We try to compare the rate of detection of MYD88 on paired bone marrow and peripheral blood samples. And what we have found is that the rate of detection of the MYD88 mutation on peripheral blood samples is lower, much lower than the bone marrow. That's why we still need the bone marrow assessment. Unfortunately, as far as we know now, bone marrow is necessary because if you analyze only peripheral blood, you risk to miss the mutation. I don't exclude that with more sensitive method, in that study that we are conducting in Italy and Spain, we will use digital droplet PCR, which is much more sensitive than allele-specific PCR that we now use in Italy and in many other parts of the world. I cannot exclude that increasing the sensitivity of the method, the things will change, and maybe we will find that also the peripheral blood is an adequate tissue to assess the MYD88 mutation status, but this is the future. This is in the future, I hope.
And I want to add one other piece to that, which is that when we've looked at previously treated patients, the sensitivity to the peripheral blood just disappears. Like it's an okay marker for untreated patients, but for treated patients, it's just, yeah, it's pretty much worthless. So and we don't know why. We have some theories, but at this point, we're still working on it. So hopefully soon, hopefully we'll be hearing soon.
That the project will give us this kind of response.
Great. Well, I think then we can a break right now, and then we'll be back soon for the next round. So thank you very much.